Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. If you like this video, subscribe my channel and click on bell icon to get notification. In this video, I am going to explain one of the topic in photosynthesis in higher plants. It is photolysis of water and photophosphorylation of light reaction in photosynthesis. The photolysis of water means the breakdown of water molecule into oxygen, H plus ion and electrons in the presence of sunlight is called photolysis of water. During photosynthesis in light reaction, at the time of electron transport, the electrons which are removed from PS2, these electrons must be replaced by the electrons released from water. And this process is achieved by the electrons which are available due to splitting of water molecule in the presence of sunlight. The water splitting complex, it is associated to PS2 only, and it is physically located towards inside of the thylakoid membrane. Water is split into H plus ions and oxygen along with electron that can be represented with this equation. In this, 2H2O is giving rise to 4H plus O2 plus 4 electrons. It is the process called photolysis of water. It occurs in the presence of sunlight towards inside of the thylakoid membrane at PS2. The protons and oxygen formed in splitting of water molecule, these are released towards lumen of thylakoid. And the oxygen which is produced in this photolysis of water as a byproduct, it is released as one of the net product in photosynthesis into atmosphere. And the electrons needed to replace those removed from PS1, these electrons are supplied by PS2. At the same time, the electrons which are removed from PS2, these are replaced with water molecule after releasing the electron with photolysis of water. During this electron transport, the phosphorylation process takes place, and due to movement of electrons in between PS1 and PS2, there is a two types of photophosphorylation. It is called cyclic and non-cyclic photophosphorylation. In this phosphorylation, it represents as addition of phosphate. If the addition of phosphate like inorganic phosphate is added to ADP, then it becomes into ATP, and this phosphorylation process occurs in the presence of sunlight and it is named as photophosphorylation. In living organisms, so they have a capability of extracting energy from oxidizable substances, and this energy can be stored in the form of bond energy in ATP molecule. Whenever inorganic phosphate is added to ADP, it becomes into ATP, and these terminal phosphate have a capability to store and hold more energy in the form of bond energy. And some of the special substances like ATP, this ATP, it carries this energy in their chemical bonding. And the process through which ATP synthesis takes place in a cell, especially in both mitochondria and chloroplasts, is named as phosphorylation. And photophosphorylation is the synthesis of ATP molecule from ADP and inorganic phosphate in the presence of sunlight in light reaction of photosynthesis. And this process of photophosphorylation, it occurs in two ways. In this, the first one is non-cyclic photophosphorylation and second one is cyclic photophosphorylation. In non-cyclic photophosphorylation, in photosynthesis, at the time of light reaction, the two types of photosystems present in thylakoid membrane like PS2, it work first and then PS1 work, and in a process the non-cyclic photophosphorylation take place. And in this electron released from water molecule, it is reach NAD plus and then it becomes into NADH plus H plus, and at this time whenever the electron from water molecule it reach PS2 and then it reach PS1 and finally release into stroma to NADP plus and it becomes into NADPH plus H plus, the entire process of electron transport take place in non-cyclic method and in the form of jet scheme. So during this non-cyclic electron transport, whenever electron travel in jet pathway, at this time that can form ATP molecule along with these NADPH plus H plus molecules, and it is achieved due to non-cyclic electron flow in between PS2 and PS1. For non-cyclic electron transportation, so both the 680 nanometers and 700 nanometer light is utilized by PS2 and PS1 respectively. And during this non-cyclic electron flow, the oxygen it is released from water molecule as a byproduct, and this oxygen it is diffused into atmosphere. And in case of cyclic photophosphorylation, some of the specialized structures called stroma lamella. So stroma lamella, it is a membrane, it present in between two granum, and these two granum are connected with stroma lamella. And in this stroma lamella, it consists of only PS1 in their membrane, but along with this NADP reductase enzyme is absent in stroma lamella. And this PS1 which is present in stroma lamella, it is responsible for cyclic photophosphorylation. And stroma lamella membrane it lack PS2. Along with that, NADP reductase enzyme also absent in stroma lamella due to absence of PS2 and NADP reductase enzyme. Stroma lamella is the main site for cyclic photophosphorylation. And the electron it is circulated within photosystem and perform phosphorylation due to cyclic flow of electron. During this cyclic electron transportation, the PS1 having reaction center with the P700, it absorbs 700 nanometer sunlight, and this reaction center get excited and lost electron, and this excited electron does not pass to NADP plus due to absence of NADP reductase enzyme, and again this electron it is circled back to PS1 through electron transport system chain. At this time, due to this cyclic electron transport, ATP molecule formation takes place. And in this cyclic flow of electron, it results in the formation of only ATP molecule, but there is no formation of NADPH plus H plus. Due to absence of NADP reductase enzyme, the electron not reach NADP plus and it circle or move in cyclic pathway, so that only ATP molecule formation take place in cyclic photophosphorylation, but there is no synthesis of NADPH plus H plus. The cyclic photophosphorylation it also occur when only light of wavelength beyond 680 nanometer light available for excitation. So to explain the photophosphorylation mechanism, many scientists propose many theories, but in this most accepted theory to explain the formation of ATP molecule in photophosphorylation is chemiosmotic hypothesis. This chemiosmotic hypothesis, it was proposed by P. Mitchell to explain the formation of ATP in both photosynthesis and respiration. So chemiosmosis, it requires some of the requirements like a membrane present in thylakoid and proton pump in the form of ATPase and a proton gradient along with ATPase structure. So ATP synthesis, it always linked due to development of proton gradient across the membrane of thylakoid, and this proton accumulation is towards inside of the membrane of thylakoid in lumen. It create proton gradient that is useful for the formation of ATP molecule. So to understand the mechanism of chemiosmotic hypothesis for the formation of ATP molecule, and during this chemiosmotic hypothesis, the development of proton gradient inside of the thylakoid in lumen, it occurred due to various steps of light reaction in photosynthesis. In this step 1, whenever the water molecule undergoes splitting in the presence of sunlight, and due to splitting of water molecule inside of the thylakoid in lumen, at this time from water molecule H plus ions are released and accumulated in lumen. These accumulated H plus ions in lumen, it increases proton concentration in lumen and compared to stroma. So it is the first reason for the development of proton gradient in lumen. Along with this, in step 2, whenever the electron it is transported through PS1, cytochrome BF and photosystem 1, at this time some of the H plus ions are transported across the membrane from stroma to lumen. At this time, the primary acceptor of electron, it is located towards outside of the thylakoid membrane, and this primary acceptor of electron, it is not electron carrier and it is an H plus carrier. That's why whenever electrons traveled from water molecule to PS2 and cytochrome and PS1, at this time whenever electrons are transported, for this electron transport frequently H plus ions are transported from stroma to lumen, and it is the second reason. So the primary acceptor of electron, it remove protons from stroma and it is transported whenever the electron transported through this structure. So these removed protons from stroma are released into inside of the thylakoid in lumen, and these create proton gradient. The third step is the NADP reductase enzyme. Generally, it is located towards stroma side of the membrane, and this NADP reductase enzyme, whenever the non-cyclic electron transport take place and electron reach NADP plus from PS1, at this time this NADP plus, whenever it receive electron, that can utilize H plus ions from stroma for reducing NADP plus into NADPH plus H plus. So due to this reaction, the H plus ion concentration gradually decrease in stroma, and compared to stroma, more H plus ion concentration or proton concentration present in lumen. That is the third reason. So because of these three steps, the protons removed from stroma and the concentration of proton or H plus gradually increase in lumen, that create proton gradient. So in chloroplast, due to these steps, the concentration of proton decrease in stroma and these protons are accumulated in lumen, that also increases pH and its concentration. And this proton gradient, which is created across the thylakoid membrane, it is measurable by decreasing pH in lumen. So due to this gradient, it is very important for phosphorylation because this proton gradient is breakdown due to releasing of H plus ions from lumen to stroma. At this time, energy released. So energy is used to pump the proton across the membrane and to create a gradient or high concentration of proton within thylakoid membrane. This gradient is breakdown due to movement of protons across the membrane to stroma through the transmembrane channels of F0 of ATPase. So the ATPase is a structure. It consists of two parts. In this, one is called F0. It is embedded within the membrane and transmembrane channel that carries out facilitating diffusion of proton across the membrane. It is called head part present towards stroma is called F1. The outer portion which is present towards stroma, it is called F1, and it is a small protrude on outer surface of thylakoid membrane on the side which is faced towards stroma. So ATPase, it has a channel, and this channel can allow the diffusion of protons back across the membrane that can release enough energy to activate ATPase enzyme that can catalyze the formation of ATP molecule. So the breakdown of this gradient can provide enough energy, and this energy causes some conformational changes in the structure of F1 particle of ATPase, and these changes helps the enzyme for the synthesis of several molecules of ATP molecules by packing energy in its chemical bond. It is the process of chemiosmotic hypothesis that explains the formation of ATP molecule. Thanks for watching.